Okay, so in this video, we're basically going to be talking about translation and how exactly the amino acids are formed from the RNA strand, which is messenger RNA. Okay, so previously we were talking about transcription, so we can't go straight to talk about translation without actually passing through transcription. So previously, we'll just have a very short recap. We have the DNA strand in the nucleus, and it is this is a molecule that is used to transcribe the new RNA molecule that eventually goes through post-translation modification and finally goes out through the nuclear pores to be translated. So this is all about transcription, where the genetic code is formed because the information on the DNA is copied onto the messenger RNA. Okay, so having said that, we'll just go straight into talking about translation. So the main steps involved in translation may be summarized under some of the following that I'm going to mention right here. We have one, the amino acid activation. Okay, the second one is where we have binding of the tRNA to the ribosome. The third one that we have is polypeptide chain initiation. Okay, the last one that we'll have is polypeptide propagation or other elongation. Okay, so actually we have another step, which is, let me just write it here. The fifth one is where we have the termination. Then finally we have post-translation. This would be post-translation modification. Alright, so having said these things, we'll first start with the binding of the mRNA to the ribosome. So I'm pretty sure you may be wondering why we haven't really talked about the activation of the tRNA. We're going to talk about that shortly after talking about the binding of mRNA to the ribosome. So I'm just going to highlight it here. So this is binding of messenger RNA to ribosome. Okay. Alright, so as we all know, we have this molecule. This is our messenger RNA, and we know that this messenger RNA is what is carrying the genetic code. Okay, so the genetic code is a series of nucleotides that carry genetic information that it previously got in during the process of transcription. So we'll start with the process. So what happens is that the small subunit, so we talked about the ribosome and we said it has a large subunit and a small subunit. So in this case, we first start with the small subunit. Okay, so the small subunit is what comes to bind on this messenger RNA. So the small subunit and the large subunit of the ribosome make they unite to make a complete ribosome. So like we said, in eukaryotes, our complete ribosome is 80S, but our large subunit is 60S and our small subunit is 40S. So these two combine to make the full complete ribosome, which is the 80S. So we'll have this happening, the large subunit and the small subunit. They have to come together. So the messenger RNA binds to the smaller subunit first. So this is the messenger RNA that's going to bind to the smaller subunit first. And after it binds, so this is our first step. We'll have the binding of the mRNA to the ribosome. To be specific, this is the small subunit. Okay? And what you'll find is that there are many clusters of ribosomes that are found on the messenger RNA. So what I'm trying to say is that when translation is happening, you won't only have one small subunit attached to this mRNA. You have many of them in the cytoplasm. Okay, so when you see many ribosomes attached to the messenger RNA, we call this a polyribosome. Okay, so like you can see from the name polyribosome, because poly means many, so many ribosomes attached to messenger RNA is what we mean when we say polyribosome. Okay, so this is what we are going to have. Like you have seen, the smaller subunit binds to the messenger RNA. So let me just draw the complete structure. So you have something like this. So this is a small subunit that has bound to the messenger RNA. Okay, so let's take, for instance, this is 40S. Alright, the next step that we're going to talk about is activation. Okay, so this is activation of the amino acids. So activation of amino acid. Okay, so before I go any further, I would just like to outline that an amino acid cannot participate in the process of translation without first being activated. So this is why this step is very important. So each target amino acid is activated by ATP. So we have ATP being involved, which is the energy currency of the cell. So this is a form of energy. This is adenosine triphosphate. Okay, so every amino acid is activated using ATP and using the enzyme. So here I'll just outline the things that we need to actually activate this amino acid. So we need an enzyme. Okay, because enzymes speed up chemical reactions. So we'll have our enzyme, which is amino acyl tRNA synthase. Okay, like you can see, just from this is amino acyl tRNA synthase. Just from the name, you can tell that something is being synthesized. So this is the enzyme that we use to activate this amino acid. So there is one enzyme for each amino acid. So there is specificity. Okay, and I'm sure you know that every protein, or rather every enzyme, is specific for a chemical reaction. So every amino acid is going to have a specific enzyme that's going to activate it. So the activated amino acid is then attached to a specific tRNA. So let me just draw this tRNA strand that looks like a T shape. So last time we talked about the tRNA having bases like CCA at the 3' end, and this is the 5' end. And these are the arms. This is anticodon arm. Okay, so what you'll notice is that here, let me just use a different color. So here at the 3' end, this is the end where the amino acid will get attached. Okay, so this is where the amino acid will get attached. So it will attach at the CAA side at the 3' end. So after this attachment, so like you have seen here, what you'll notice is that we'll have an amino acid attached here, and the tRNA complex is ready to transport the amino acid out of the ribosome. So when this amino acid gets attached here, we call this entire atom, or rather entire molecule of the tRNA, it will be referred to as, it will be called activated. Okay, it's activated because it has an amino acid that's actually also activated, and this is the tRNA that can actually participate in the process of translation. So what we have here, let's go over the steps. We have ATP involved, we have the enzyme, which is amino acid or tRNA synthase. We have we said there's always a specific enzyme for a specific amino acid. Okay, so let me just summarize all this with an equation. That way you get a better understanding. Alright, so when you look at this, so we're going to have, the first thing is our amino acid. Okay, and like we said, we have ATP that helps to activate this amino acid. Now, having said that, for us to get our products quickly, we need an enzyme, which is amino acyl tRNA synthase. Okay, and our products will be the activated amino acid plus AMP. So don't get worried about this AMP. This is adenosine monophosphate. So I can actually share more about the chemistry of what is happening here. Okay, so what you'll find out is that ATP is a very high energy containing molecule, such that when you break it down, we'll have this high energy phosphate group or phosphoryl group plus the adenosine monophosphate. So it is this phosphate group that when it is released, there is energy that's involved. So this is the energy that's going to help activate this amino acid. So the amino acid plus the ATP under the amino acyl tRNA synthase is what forms the activated amino acid. And like we saw, let me just write the next equation so that we can appreciate what's actually going on here. So now that we have our activated amino acid, the next thing, so our abbreviated amino acid with just 2 lowercase a and another a. So the activated amino acid plus our tRNA okay, so this is the next step. So what we'll note is that this tRNA will now get attached to our activated amino acid, and this is what makes the tRNA actually an activated tRNA. So our amino acid is going to be here at the 3' end. Okay, now we have an activated amino acid. So this is tRNA and this is amino acyl tRNA. Okay, so when you see amino acyl tRNA, just know that it's activated, an activated tRNA. Okay, next up we are going to talk about, we're actually going to talk about the polypeptide initiation. Okay, okay, so when something is being initiated, you automatically know that there is a beginning of something or a process. So under initiation, this is where we're actually starting to, or let me just say the starting point of TR of translation. So what you note that the tRNA with the anticodon that we've been talking about, okay, So this is our tRNA. This is our anticodon. Okay. So the first anticodon is UAC. It forms the codon AUG. Okay. Now let me take you back to binding of the ribosome to the small subunit of mRNA. So remember, we have a small subunit. Okay, we already have this small subunit that's already bound, and this is our activated tRNA. Okay, with an amino acid there. Let me just write it nicely. Okay, with the amino acid right here, depending on what the 3' end is. Okay, so this tRNA is going to come and bind here. Okay, with its amino acid. Now, what I'm trying to say is that on the messenger RNA, the bases read AUG, but on the anticodon it reads UAC, because remember, these are complementary. So if in the messenger RNA you have adenine, in the anticodon you have uracil, and so on and so forth. And every tRNA reads the bases 3 at a time, because you know these are codons. Okay, so all these are codons, and they code for information that was gotten from the DNA during transcription. Okay, so the tRNA is going to bind on this messenger RNA that's bound to a small subunit of the ribosome. And the first amino acid, okay, so this is very key. The first amino acid to be translated is AUG, which stands for methionine. Okay, so you note that we had spoken about the genetic code and how you can have more than one codon coding for one amino acid. But when you look at this AUG and tryptophan, these are the only bases that only have one codon for one amino acid. Okay, and this is very cardinal, because if you had many starting points, there would be so much confusion during the polypeptide synthesis. So we only have one starting point, which is the AUG, which is denoted by the methionine. So what I want to say is that this activated amino acid that you can see, it is like an empty amino acid, okay, though it's activated. So what gives this amino acid identity is the information that is read by the tRNA that has been coded on this messenger RNA. So when the AUG is read by tRNA, automatically this amino acid now gains identity and becomes the methionine that you can actually see here. So this is to actually show you that this amino acid that was activated, it doesn't come here as methionine already. What determines its identity is the genetic code right here. This is what to determine what type of amino acid it's actually going to be. Okay, so the AUG is the messenger RNA start codon, like we said, and the anticodon is the UAC or FMET. Okay, so the anticodon on the FMET tRNA is this UAC. Okay, so proteins code initiation factors are involved in this synthesis, or rather initiation. So we have what we call the protein factors, though we'll not really dwell so much on that. So these are protein factors. These protein factors actually just helping during the initiation process. Okay, so this is what is happening. So let me just draw a quick diagrammatic drawing that you can just see how everything is happening. So we have the activated tRNA, okay, plus the messenger RNA that's bound to a ribosome, the small subunit. So when these two come together, this is how initiation takes place. Okay, so we're going to have this happening. Okay, this is a small subunit, so they're bound. Now what you'll notice is that when the tRNA binds to the small subunit, only then can the large subunit bind. Then this is the full process of initiation. So this is the large subunit. Okay, large subunit. This is the small subunit. Okay, so we're going to now go in chain elongation. All right. Okay, so we're here. We're now talking about chain elongation. All right. So what happens is that we already have our tRNA molecule that is bound to the messenger RNA with its amino acid, and this is our large subunit. Okay, and it's also bound to a small subunit, and this is the messenger RNA that's being read. So what you notice is that for chain elongation, this is simply us trying to make the amino acid sequence. So you notice that this amino acid chain, it will keep growing. The more we have more tRNAs coming into this this ribosome for protein synthesis. Okay, so we have an amino acid. So what we want to say is that the second tRNA that is coming here with its amino acid, okay, so this is just tRNA trying to bring another amino acid. It's going to come and bind in what we call the A site. So this is the A site. Let me just highlight it and give it a different color. So this is the A site. Okay, so we call this site where the new incoming tRNA comes to bind. Okay, so it comes in the A site, and this is what we're going to have. Okay, so follow each step with caution. So this is our first tRNA, okay, with its amino acid, and our second tRNA has come with its amino acid. Okay, so what you notice is that when this second amino acid comes, the two amino acids are going to bind. Okay, so this site where the first amino acid was, we're going to call it the peptidyl site, because this is where a peptide bond is formed. Okay, so what you notice is that this is our mRNA. So what you notice is that every time this small subunit moves to the right or to the left, depending on how it's initiating this, when it moves one codon to the right, it's going to leave an empty site. Okay, so if the messenger, if this complete ribosome was here, it will now move here. Okay, this is our new, so it means this ribosome that was in the A site. Okay, so let me just label them. This is P, this is A site. So the ribosome, the tRNA, sorry, that was in the A site, immediately this ribosome moves to the left. Okay, this tRNA will now find itself in the peptidyl site. Okay, so this is to tell you that the two amino acids will remain attached to it. Okay, and because the ribosome has moved a codon ahead, it means this amino acid, let me just point it, this amino acid will now be in an empty space that we're going to call the E site, or rather the exit, and from here it will just leave, okay, because this site is empty, so it will leave. So the next thing will happen, the second amino tRNA will come here with its activated amino acid. It's going to come and bind here in the A site, okay. So this is it coming. So let me just draw another one and show you how this elongation is taking place. So this is the same thing. I'm just trying to project what will happen. So now that the tRNA binds on the S site, okay, so this is, let me actually use different colors so that you're able to appreciate what's happening. Alright, so these two amino acids, okay, remember that this one had a different amino acid attached to it. It had two amino acids, sorry. So what will happen is that these two amino acids will get attached to the third amino acid, okay. So remember, this is the A site. The same thing that happened in the previous one will happen. The ribosome will move a codon ahead, so it will come and read. So before we go further, I want you to know that every time a tRNA comes in the ribosome, it always reads the information on the messenger RNA and projects it on the amino acid that it has and gives it the identity. So this could be serine. So this amino acid sequence, it's composed of different types of amino acids, not just the same type. So you can have serine, you can have methionine, which is always the first one. You can have tryptophan, and so on and so forth, as many as you can. So initiation is always taking place like that. So you find all these, that there's a genetic code. So let me write it here. So if the first genetic code was AUG, the second one can be UAC, the other one can be CUG, just like that. So you find the tRNA is actually reading these codes and projecting them there, just like that. Alright, so the third amino acid is brought to the S site, okay, just like what happened here. The third amino acid will be brought to the S site. So let me just use a pointer. So the third amino acid will be brought here, okay, to the S site. So as this ribosome moves a codon ahead, it will leave this ribosome that was being used in the, to leave it in the E site, and it will leave, okay. And as this one is leaving, it leaves all the amino acids it was attached with this amino acid that's in front of it, which is the one in the A site. And immediately it does that, the one in the A site now will become the one in the P site, where the peptide bonds are being formed. And so the chain continues, and so on and so forth. Okay, so we can come to our other stage, which is chain termination. Okay, so under this process, whenever we have UAA or UAG or UGA, immediately this ribosome, okay, we have this ribosome complex with the tRNAs, okay, immediately it encounters any of these. I'm not saying they should be encountered all at the same time. As long as it meets any of these, that means that translation is over. Okay, so these are referred to as stop codons. Okay, they signal that the process is done, so they're called stop codons. Okay, so whenever they reach the S site, okay, or rather the active site, the polypeptide must fold. So this polypeptide chain that was being formed is going to fold within itself. So it will form something like this. And always remember that the new polypeptide chain that was being formed is just going to be within the walls of the ribosome. It will not go out, it will just be within the walls of the ribosome, and it folds itself as they approach this UAA, UAG, or UGA. Okay, so the synthesis of the polypeptide chain stops, and at this point the polypeptide chain is released. So whenever this is encountered, this new polypeptide chain is now going to be released into the cytoplasm to be further folded in the Golgi apparatus, okay, which you are going to learn with time. Okay, so this Golgi apparatus is where the proteins are now folded. It's like a factory where we now get finished products. So this is just a sequence of amino acid, but in the Golgi apparatus we will now have a proper amino acid. If it's going to be hemoglobin, if it's going to be the proteins found in your hair or your nails or anything, or enzymes in the stomach, this is what determines the Golgi apparatus. Okay, and immediately the polypeptide leaves, so the polypeptide will exit. What happens is that the ribosome will detach. So the small ribosome will detach itself, and even the large subunit of the ribosome, they'll all detach themselves, and the messenger RNA is also detached. So this messenger RNA will also go away. It is actually degraded. Okay, so in readiness, so all this is happening, and this will be the end of translation. So the next thing we'll talk about is actually the post-translation modification. Okay, post-translation modification. Okay, so when you look at this, in order to achieve a biologically active form, the new polypeptide that we talked about. Okay, so let me just take this. This is our polypeptide, a long chain of amino acids. Okay, it must fold itself. So folding is like if I'm sure you learned about biomolecules and you learned about proteins, and you must have learned that each and every one of them have a chemical structure. So there is a way in which they fold around themselves such that others will be hydrophobic, others will be hydrophilic, others will be, they'll have sulfur inside. So this is protein folding. So before or after the protein folding, we have, they must undergo enzymatic processing. So this folding is for this protein to form a 3D structure. Okay, a 3D structure. Now this structure can be an enzyme, to be specific. It could be the any protein in the human aspect. Okay, so the first thing is that it has to be processed before this can be folded. Before it's folded and after it's folded, it needs to be processed. Firstly, by the removal of amino acids. Okay, so we need to remove amino acids that are not functional. We need also to, so let me just write it in a stepwise manner so that you're able to follow through. Okay, so the first one is removal of amino acids. 2, you have addition of functional groups to the certain amino acids. So addition of functional groups. So this functional group is what determines the nature of an amino acid, because remember an amino acid has an alpha carbon. Okay, it also has an R group, it has a hydrogen molecule, a carboxylic group, and an amine group or an amino group. So this functional group during post modification, during post translation modification, on the R group, we're supposed to add a specific R group that would determine the uniqueness of an amino acid. Okay, so if you add an acid here, it means overall this amino acid is going to be acidic. Okay, because this amino group and the carboxylic group, when they are together, when you mix an acid and a base, they will form something neutral. So it means if the environment is neutral, whatever you add to that environment becomes the final condition of the amino acid. Okay, it's like water. If you add an acid to water, the water will be acidic. If you add an alkaline to that water, it's going to be alkaline because overall it's neutral. Okay, so this is what we mean by adding the functional group. Okay, thirdly, we have proteolytic cleavage. Okay, so let me just erase this. Okay, so thirdly, we have proteolytic cleavage. Okay, and attachment. So don't worry, I'll get back to the third step. And attachment of oligosaccharides. Okay, so this is our last step. So insulin is an example of a polypeptide which undergoes such a type of modification. It is synthesized as insulin, but again it must be modified. So this is our insulin. It's one of the examples that we can actually take. So initially when it's synthesized, it's a very long molecule. So we said some amino acids have to be removed. Okay, those which amino acids don't code for the making of the insulin, they'll be removed. Next thing, addition of our functional group. Okay, of our functional group. So if I when we remove, the chain is going to now reduce. It will not be as long as it was. So we've now added a functional group. So the functional group here is this in green. Next, we have proteolytic cleavage. Okay, so to cleave something is where you just cut it. So this is going to be cleaved, sorry. So if this is being cleaved, you have something like this now. It will have two parts. Okay, one that we can call the alpha chain, the other one that we can call the beta chain, because this has been cleaved. Okay, and then we'll have attachment of oligosaccharides. So the different types of proteins, there are those that we call the glycoproteins. So if it's glyco, it means an oligosaccharide was actually added on. All this is done during modification. So you must know that it's not all amino acids that can have an oligosaccharide attached to them. Okay, because not all proteins are glycoproteins. Another example of a polypeptide that can undergo post-transcription modification is trypsin. Okay, so we have another one, it's called trypsin. It can undergo such a thing. It is synthesized in an inactive substance called trypsinogen. Whenever you see where they've added an inogen, you see something like this, just know that this is inactive. Okay, so the active form is going to be called the trypsin. The inactive form is what we call the trypsinogen. Okay, so this is all about post-translation modification. Alright, so this is the end of translation. So let me just give an overview summary of everything we have been talking about? So the first thing that you have to note is binding of ribosome to mRNA. The next thing that you note is that the amino acid has to be activated. So activation of amino acid, okay? And you know that during this process, even the tRNA is going to be activated. The next thing is that this activated tRNA binds. So we have binding of tRNA, to be specific, activated tRNA to ribosome, to the ribosome complex. Okay, so I'm calling it a complex because this ribosome is attached to messenger RNA. After this happens, we'll now have the initiation. Okay, so initiation, you have what we call elongation. Then elongation, you have termination, which we talked about. And the last thing is that you have post-translational modification. Okay, so these are basically the steps that are involved in translation. So this is the end of the video. Feel free to give reviews on it and let us know what we can work on, and so on and so forth.